Sir, it's allowing us to ask a question, but I don't think you can like chat.
Okay, can you just ask question if I want to see if that works, how it works, because today is the first session. Trying to set it up. So it's not working, perhaps. So I don't know how you will be asking questions. If you have any doubts, probably you have to unmute yourself and do that. So let's begin. So I am Sanket Mukherjee, a PMRF fellow from IIT Delhi, from Kusuma School of Biological Sciences. Hopefully all of you are enrolled to this NPTEL course, Genome Editing and Engineering, uh, which is taken by Professor Utpal Bora of IIT Guwahati. I Just to give you a brief introduction about myself, I did my bachelor's in BTech. I mean, bachelor's as BTech in biotech from Kolkata. Then I did my MTech from IIT Guwahati itself. And then I'm pursuing my PhD here at the School of Biological Sciences, IIT Delhi. So this is a 12-week course. Just give me, yeah. It's a 12-week course. And every week we will be meeting like this every Tuesday on this time 7 to 9, and we will be discussing, ideally the summary of whatever has been taught in that particular week, and then we will discuss few assignment questions, few sample questions basically, which will help you to solve the assignments. So are the annotations visible to you, all of you?
Yes, sir. The annotations are
Okay,
visible.
fine. So I am assuming that most of you have a background where you have molecular biology as one of your subjects in either your undergraduate or postgraduate degree. And if you are aware of it, then you will be it will be a little bit easier for you to know the terminologies. But even if you're not, it's all right because I'll be telling you what are the main terminologies that we'll be using throughout the course. So one of them is gene. So gene is a basic unit of heredity, right? And uh, it's present in the chromosome, in the nucleus of a cell, and essentially it encodes for proteins. And these proteins are very important. They have a functional role later in your in various physiological processes of a cell. Now, when these set of genes, right, these set of genes in an organism is called a genome, right? It's called a genome. And when we talk about genome editing tools, what we mean is that these tools can be used to edit genes, um, and the entire genome of an organism. We will see what are the tools, how to go about it, and other things. So another basic thing which I hope a lot of you know is the difference between a prokaryotic and a eukaryotic cell. So there is a list which is already given in the lectures. But what is important from the genome organization perspective is that in prokaryotes, the gene, or the genome rather, is not localized in a compartment. It is localized at one part of the cell, but it is not a membrane bound compartment. It is called the nucleoid, right? On the other hand, there is a specific cell membrane inside the cell, in case of eukaryotes, which contains this genome of the organism. And there are different processes which occur, like there is replication and transcription and translation, things like that. So we'll talk about it. But the thing is, the genome organization of the genomes of prokaryotes and eukaryotes are different. That is the idea from this slide. Now, earlier, it was in historically it was not known that what is particularly the element in a cell that is transferred from a parent cell to a daughter cell, or something like that. So for that, there were various sets of experiment. One of them was the Griffiths experiment. So what happens here is, there is a bacteria which has two strains. Either it is the rough strain, which is non-virulent. So non-virulent strains will not kill or will not have the pathogenic function, whereas the smooth strain were virulent strain. So when rough strain was given to mice, the mice dies, as you can see in the first column. So if if the rough strain is given, the mice lives. If the smooth strain is given to the mice, injected to the mice dies, right? And what was interesting to observe was that if a heat-killed smooth strain was given, so if through heat inactivation, if the smooth strain was killed, then that was not able to kill the mice. So this is the third column. And another interesting thing was, if rough strain was mixed with this hit inactivated killed smooth strain, it was observed that the mice dies this time. And this was contrary to the expected observation. So Griffith said that there is some transforming principle that could, change a rough strain into smooth strain, right? And this started the journey of discovery of the transforming principle. What is the transforming principle? Now, based on this, further experiment was done on Avery, McLeod, and McCarty. These three group of scientists, they did further experiments. So essentially, in a cell, there are three main components which were thought to be, you know, passed on from one generation to another generation. So the nucleic acids, like the DNA and RNA, are one of them. Like, these are the two factors which could be passed on from one generation to another generation. Another thing which was thought to be involved was protein, because protein majorly has a lot of functions. It majorly has a lot of functions in your body. Proteins are the main factors. So it was also considered to be one of the major things which could be transferred. Now, in this experiment, what they did, they took virulent S strains, they killed these strains, and then they added either DNAs, RNAs, or proteinase. So these are enzymes which can hydrolyze. So DNAs can hydrolyze DNA, RNAs can hydrolyze RNA, and proteinase can hydrolyze protein. So that is the idea basically. So when they added protease, so in one tube they added protease, in another tube they added RNAs, in another tube they added DNAs. So when they added protease, there was no protein in the mixture, right? It was hydrolyzed. When RNAs was added, there was no RNA in the mixture. When DNAs was added, there was no DNA in the mixture. And after this, they added the rough strain. They mixed the rough strain and they wanted to see which particular component is successful in transforming the rough strain to smooth strain. And to their finding, what they found was that wherever DNA was present, DNA was not degraded. Then in those tubes or in those falcons, they could see that there was transformation happening. DNA, when it was removed using DNases, then this transformation was not happening. So DNA came out to be the transforming principle. Now, another experiment, very similar experiment, was conducted. So these are some series of experiments. It's already there in your slides. So here, bacteriophages were used. So the Hershey and Chase experiment. So bacteriophages are viruses which can infect bacteria only. So they labeled the DNA or they labeled the protein, right? So this is called radio labeling so that it can be detected in the cell. So once they radio labeled the DNA and then they radio labeled the protein, and they allowed the bacteriophage to infect the bacteria, and after which they centrifuged the mixture so that the bacteria and the bacteriophage can be separated, and they found that in the cell mixture, DNA radiolabels were found. So it was it is another further confirmation that DNA is the material that gets transferred, right? So then we come with to the DNA structure. All of you know about the structure of DNA, I hope so. So before that, we need to know about Chargaff's rule, which states that the number of adenines is equivalent to the number of thymines, and the number of guanosine is equivalent to the number of cytosine in a DNA. Essentially, A plus G will be equivalent to T plus C. This is what Chargaff's rule is, or it can also be written as purines are equivalent to pyrimidines in DNA, right? So this is what Chargaff's rule is. So using X-ray diffraction image of the DNA, they were able to find out the helical structure of DNA, where A and T they form double bond, and G and C they form triple bonds. The structure of DNA is very important for you to remember. And important thing about this is there are two antiparallel sheets. So one is from 5' to 3', as you can see here, 5' to 3', and then the other one goes 3' to 5'. So this is how DNA looks like. We will be talking about DNA very often. Now, from this DNA, any questions so far? You can raise your hand if it is all right for everybody, if things are going well. Am I audible? Yeah, okay. Thank you. Fine. Then we come to the genetic code. So what is the genetic code? So we know about DNA. Now DNA is made up of 4 letters, A, T, C, and G. So it's pretty clear to everybody. Now this combination of this A, T, C, and G forms the entire DNA. Whatever is present in your body, in the cells of any other organism. So in any case, basically. So the question arose from this is, there are how these 4 nucleotides could code for numerous proteins, right? That was a question which came up. Now, proteins in general are made up of amino acids. I hope you know this. Amino acids, it's made up of amino acids. Now, there are 20 amino acids. There are 20 major amino acids. Like there are 4 different letters for DNA. For amino acids, there are 20. And these combination of amino acids, they form protein, right? Polypeptides and then proteins. So how do 4 particular, I'm sorry, how do 4 letters code for 20 amino acids? So that was a question which came up. And through the research of Hargobind Khorana, we found that codons are involved behind this. So in triplicates, like if you see in this table, let's say G, A, and A, it codes for a particular amino acid, glutamic acid. So this is the codon, it's the genetic code, this is the table of genetic code, which tells you which triplet, if a DNA is in a particular sequence, 3 letters will correspond to which particular amino acid. And something very interesting comes up from here if you observe that that the 3rd position of this genetic code, this codon, is not that relevant in terms of the amino acids, and that's why it's called the wobble position. And then another thing which we see from this table is that 61 codons, they they code for 20 amino acids, and the rest 3 codons, they are stop codons. So if these 3 codons are present, which is basically UGA, this one UGA is one stop codon, UAA and UAG. These 3 are the 3 codons which are stop codons. If these codons are encountered, then there is no further amino acid which is synthesized. Now coming to the genome organization of let's talk about prokaryotes and then eukaryotes. So in case of prokaryotes, mostly the genome or the DNA is essentially, it's circular. Now this circular DNA is usually in a supercoiled form. And if you see a cell like this, if this is a cell, it's a bacterial cell, let's say. So there is not a particular membrane that is separated for this arrangement of genome. So there is no membrane, but this DNA, it remains in its supercoiled form in one location, and that is where the genome is, right? And there is origin of replication in this genome, and from this origin of replication, there is replication. So I hope you know what is replication and translation and transcription. You know these things, all of you? If anybody doesn't know, they can ask. I'll still tell you. So the central dogma of molecular biology, so it tells that DNA, it forms more DNA using replication. This DNA forms RNA through transcription, and this RNA forms proteins through translation, right? So this is the central dogma of molecular biology. So when we talk about replication, it's basically DNA making more DNA, right? So we talked about the genome organization of prokaryotes. Let's talk about eukaryotes. So in eukaryotes, there is a huge amount of DNA that is present, right? And it is highly supercoiled using some proteins, which we call as the histone proteins. So these histone proteins, they are positively charged proteins, and the DNA is negatively charged. Now a DNA is wound across the histone proteins. so that it can be compacted into the nucleus of the cell, right? That is the idea. So this is how it is. Let's go. Okay, yeah. So here nucleosome is a particular, it's a histone molecule with DNA wound around it. So around one histone molecule, a DNA is wound around 1.65 times, and histone, they form octamers, right? They form octamers, and that is how the organization is. Now, talking about genetic engineering, these are some lab-based techniques to alter the DNA of an organism. So when we talk about altering DNA, this is the essential, factor, which is basically which is the code of the entire cell, right? This if you look at it uh, as a computer, then DNA is the source code that runs the computer. So altering of this DNA is genetic engineering, and Paul Berg was the first person to create the first recombinant molecule. He created the first recombinant DNA molecule. So there, apart from him, there was Herbert Boyer and Stanley Cohen. They were able to create transgenic organism. So transgenic organism means that ideally that organism doesn't have this gene, but you introduce this gene from some other organism. So they were able to create antibiotic resistance. I mean, insert the antibiotic resistance gene in E. coli. So the details are there in the slides. I'm just skimming through the important topics. Uh, Now, the basic steps which are there in genetic engineering is firstly identification of the gene of interest. So let's say, for example, if you want to have this, let's say if you want to have this antibiotic resistance gene inserted, so you need to identify that which particular location and which particular gene in the bacteria it has this antibiotic resistance. So identification is the first step. Then you need to isolate it. And then there is cloning. So how is cloning done? For cloning, there is an insert. So this is, let's say, an insert. And then there is a vector. So both of these are DNAs. Vector has the ability to, self-replicate in a bacterial cell or even in an animal cell. It could happen in either of them. So it's usually a plasmid mostly when we perform cloning. So what we do is we perform restriction enzyme digestion. So restriction enzyme digestion would digest the insert as well as the vector. So it will digest both of these things together. And then what we go for is the SIP treatment of, or that is alkaline phosphatase treatment of the vector, so that the vector does not self-ligate. And then we incubate these vector and insert together in the process called ligation, where the insert is now inserted into the vector. So this is the concept. Basically, this is the concept of cloning. Now, once clone is made, this clone is inserted into the host genome. So for case of bacteria, we perform transformation. For animal cells, we perform transfection. There are various methods to, insert this foreign DNA particle inside the cell. So we'll discuss that also later. And then the expression of this target gene, because once you insert this gene, you want to see if the gene works, right? So These are the basic things. So genetic engineering tools, as we already see, the two major tools are the enzymes and the vectors. So these two are the main tools that we will be discussing about even today in this class. So the restriction enzymes, they cleave DNA fragments at or near a specific site, right? That is what restriction enzymes do. So I hope you have studied about it already in the lecture. And it was discovered by Werner Arber and Matthew Messelson in the 1960s. So they were studying about type 1 restriction enzymes, right? And in 1978, the group, so Arbor and then Daniel Nathans and Hamilton O. Smith. So Smith, they received the Nobel Prize for this discovery of restriction enzymes. So restriction enzymes could be also of two types. Either they could be exonucleases. So exonucleases, they cleave the DNA. So if this is the DNA, they cleave from the terminal. So from the terminal ends, either from the 5' to 3' end or the 3' to 5' end. So this is how exonucleases work. Endonucleases, on the other hand, which is more used in molecular biology. So endonucleases, they recognize the site, and then they cleave it either at this recognition site or a little bit farther away from the site. So it is based on the enzyme which you are using. Most restriction enzymes which are used are, do you have a question?
Hello, sir. Sorry to interrupt. Are you sharing this? Like, have you shared this presentation already? Because
No, I have not, but I will be doing it. Along with the annotations, it will be there.
all right,
So
sir.
yeah. So there will be a drive link for it, and the NPTEL team will be sharing this with you.
Okay, thank you,
So
sir.
yeah. Hmm. So I was talking about recognition site and endonucleases. Yeah, the most frequently used type of restriction endonuclease is type 2P, that is type 2 palindromic restriction enzyme. For example, there is EcoR1, EcoR5, BAMH1, and there are multiple restriction enzymes of this category. So what does palindromic mean? It means that the sequence of this restriction, I mean the restriction sequence reads same from 5' to 3' end as it reads from for 3' to 5' end. So for example, for EcoR1, the restriction site is GAATTC from 5' to 3' end. And if you do the same thing from 5' to 3', it will again read the same. So this is an example of this restriction enzyme. Okay. Then what we need to know, so these are the restriction enzyme types. So type 1, which was first discovered. So it's a bifunctional enzyme which methylates and has an endonuclease activity as well. There are three different subunits for type 1 restriction enzymes, and ATP, magnesium ions, and SAM, S-adenosine methionine. These are the three cofactors which are required. Magnesium is always a cofactor for all restriction enzymes, regardless of the type. And the cleavage site of type 1 restriction site is 1000 base pair, around 1000 base pair away from the recognition site. So this is about type 1. Type 2, which is the most used enzyme in molecular biology, is unifunctional, which either has endonuclease activity or methylase activity. Now they have two identical subunits, and they also have magnesium ion as cofactor, and they cleave at the recognition site. Okay, so right where there is recognition of the sequence, there only there is cleavage. And in type 3 restriction enzyme, these are bifunctional with both endonuclease and methylase activity, which has two different subunits. And there is ATP and magnesium ions, which are cofactors, and they cut around 24 to 26 base pair downstream of the recognition sequence. So can anybody tell me why? I mean, restriction activity or endonuclease activity is very important, but why is methylase activity also present in case of type 1, type 2, type 3, all the restriction enzymes? So can anybody just share if they know? Yes, no? Okay, if you don't, so usually restriction enzymes are strategies which are developed by bacteria if it is found isolated from bacteria. So it's a strategy by bacteria to restrict the entry of any foreign DNA. So if any DNA which is not of the bacteria's own, it will restrict it. That is the work of these restriction enzymes, ideally in a cellular setup, right? Now, these restriction enzymes, it is possible that these restriction enzymes could also cleave this DNA of the organism itself, like it could cleave the DNA of the bacteria itself. To prevent that, all these recognition, the same restriction enzyme, they have a cognate methylation activity. So what it does is it methylates the own self DNA, so that once this DNA is methylated, it is no longer recognized by the restriction enzyme for restriction. So it prevents its own DNA's degradation or restriction. But if there is a foreign DNA that comes in, then it essentially cuts that DNA. So that is how it works. So any restriction enzyme, it produces either sticky ends or blunt ends. So for example, for sticky end, we have EcoR1, we already discussed. So it's GAATTC, and again, the same read backwards. So it cuts after G, and this produces sticky ends, right? That's the idea. Now, if you look at blunt end restriction, so it is the example is EcoR5, that is GATATC, again same, and it cuts after this T. So blunt ends so a restriction enzyme could be either sticky end or blunt end. So these are the concepts of sticky end and blunt end. Then the enzymes could be isoscizomers, neoscizomers, or isocodamers. So isoscizomers, they recognize and cleave identical sites. For example, there is SPH1 and BBU1, which recognize CGATA. CG. So this is the specificity. This recognition, this restriction enzyme could recognize C's and G's at equal proportion, and they can cut it. So this is like these two restriction sites, they have identical cleavage and recognition sequence. Neoscizomers, they recognize the same site, but they cleave differently. So for example, SMA1 and XMA1, these are two different restriction enzymes which recognize the same site, but they cleave differently. So if you look at, SMA1, the recognition site is GGGCCC, and SMA1 cuts here in the middle. It's a blunted and end cutter. If you look at XMA1, it will be the same sequence which it recognizes, but it cuts right here after the first G. Isocodamers, they recognize slightly different sequence. But upon cleavage, upon the restriction, they produce the same product. So for example, here MBO1 and BAMH1, these two are the restriction enzymes. So for MBO1, it is NGATC. This is how it cleaves. So this is the restriction product. For BAMH1, it is GGATCC. Same here. So the restriction product is same, but these are two different enzymes. Apart from this, there are other enzymes as well. So as we already talked about DNA ligase or DNA ligases in general, they are used for, joining DNA nicks. So it catalyzes the formation of phosphodiester bonds in a DNA. If you remember the DNA structure, it will be easy to know what phosphodiester bonds are. Whenever there is a single-stranded break, DNA ligases join these breaks. Another enzyme which is important is polynucleotide kinase, also known as PNK. Polynucleotide kinase, they cause phosphorylation of DNA. So if the ends, basically if the ends of DNA, they are hydrolyzed. So if let's say this is OH and OH in the 3' end, so this kind of DNA, it cannot essentially ligate because phosphate groups are important. So upon treatment with PNK, this will have a phosphate group on the end. So that's how it is used. Another enzyme, very important enzyme, is alkaline phosphatase, which has the opposite role of the polynucleotide kinase. So alkaline phosphatase, it removes in a DNA, always at the 5' end, there is a phosphate group, and on the 3' end, there is a hydroxyl group, right? Now, it's always there in case of DNA. It's double-stranded, so it will be like this. This is 5', this is 3', right? And there are complementary bonds. Now, an alkaline phosphatase will remove phosphate from the 5' end. So these phosphates will be removed. It will basically be like this. So as I already told you, CYP treatment, so it's basically alkaline phosphatase treatment given to vectors, usually during cloning, so that the vector cannot self-ligate together. So sometimes it is important to remove the phosphate groups if we do not want some unwanted products to be formed, right? Some alkaline phosphatases are well known based on their sources, like the bacterial alkaline phosphatases. Then there is the calf intestinal alkaline phosphatase. And then there is the shrimp alkaline phosphatase. These are the very common alkaline phosphatases which are used regularly in molecular biology labs. So I hope it's clear till here. Okay. Okay, let's talk about vectors. So vectors are DNA. Okay. Sorry. Yeah. So vectors, these are DNA molecules that can carry your DNA of interest, right, inside the host cell for cloning. So the main two important features for a vector is it should be able to replicate itself. So replication and then expression. These are the two main things that are required in a vector. So the most used vector is a plasmid. I hope you know about plasmids. These are small extra chromosomal, DNA, which are usually found in bacteria, right? There are certain characteristics of a plasmid, which we do want for our molecular biology experiments or genetic engineering experiments. So one of the characteristic is it should have an origin of replication so that the plasmid could a cell. So that is one of the characteristics. Another thing is there should be a selectable marker. So a selectable marker usually most of the time which is used is antibiotic resistance. So for example, you have inserted a particular gene inside inside a bacteria, for example. Now, how would you know that the bacteria, the colony, is actually having that particular gene of interest or not? So to select that, we use these selectable markers. Mostly it is antibiotic resistant markers. So for example, if you in the vector, there is a ampicillin resistant gene which is already present, then if you grow the bacteria in presence of ampicillin, only the ones which contain your gene of interest will be able to grow. So that is the idea. The third one is it should be small in size because if it is very big in size, it is difficult for the cells to take in. That is one thing. It should have multiple cloning site. So basically, multiple cloning site means it should have, it should be accessible to multiple restriction enzymes so that you can make the most use of these vectors. And then another thing is should be easily isolated from host cell. So if you cannot isolate it, if it gets integrated into the host cell, then it becomes a problem. So these are the main characteristics of a plasmid. So after this, we have the difference between a cloning vector and a expression vector. So ideally, in a molecular biology setup, when you target a gene, there are two reasons why you target it. Either you want to have multiple copies of this gene, right? That could be one idea. Another one could be that this particular gene encodes for a particular protein and you want to have this protein. For example, in case of insulin, so you want to target the insulin gene and you want to produce the protein so that the final protein is your interest, your product of interest, basically. So if you want to have multiple copies of a gene, there are different sets of vectors which are used, which are called cloning vectors. So these are short pieces of DNA that introduce a foreign gene into a host cell, and it is stably maintained inside the cell. And it is usually used to maintain multiple copies of a target gene. It contains the origin of replication, selectable marker, and restriction sites. The examples are like plasmid, cosmid, BAC, YAC, we'll talk about it. These are bacterial artificial chromosomes and yeast artificial chromosomes and phages. For expression vectors, this is a vector which the idea behind this vector is not to create multiple copies of the gene, but to express the gene so that you can have a lot of protein, right? That is the idea. So it contains the promoter, enhancer, start, stop codon, selectable markers again, origin sites and restriction sites. So for example, a PGEC series of vectors, these are used for the expression studies. So then we have some more, terms like shuttle vectors. Shuttle vectors are mostly used for, when a single vector, which could be a plasmid, can propagate in more than one host. For example, there are PGL3 group of vectors. These are some artificial plasmids. PGL3 group of vectors. Then there is, Agrobacterium tumefaciens. That is also, it has its own vector, which is able to, propagate in bacteria as well as plant. In case of PGL3, it can propagate in bacteria as well as animal cells. So these are called shuttle vectors. Bacteriophages or simply phages, they only infect bacteria. These are viruses, basically. These are viruses that only infect bacteria, right? And we use them because when they infect bacteria, they transfer their genetic material. So we use them to transfer our own genetic material, our genetic material of interest, basically. So there are two bacteriophages mostly known. That is the lambda phage and the M13 phage, right? And one of the advantages, major advantages of using bacteriophages is it can, insert large size of DNA, or we can use large inserts, basically, in case of bacteriophages. A phage mid is a combination of phages and plasmid. So that is, if you want to have the best of both these things, it's called a phage mid. And a cosmid is it's again a plasmid with more than one uh, lambda phage cohesive sites. So this is called cosmid. Well, then there is yeast artificial chromosome. Or bacterial artificial chromosome. The main reason for these artificial chromosomes are they can insert a very, very large, amount of DNA. If you're talking about DNA, like in case of humans, so for cloning, a very large segment of DNA, in case of human DNA, for example, you could use a yeast artificial chromosome or a bacterial artificial chromosome for that. The disadvantages of this is that whenever you increase the insert size, these plasmids or these vectors, they become less stable, and the cloning efficiency. So the cloning efficiency is basically once you are trying to make a clone, what are the percentage of that mixture which will be positive, which will have the positive clone. So the cloning efficiency usually is low in case of these large artificial chromosomes. Another uh, is Agrobacterium tumefaciens, which has the TI plasmid. This TI plasmid has transferred DNA, which is transferred into the plant cell. So that's what it is about. Then let's move on to the next slide. Okay. Why is it not moving? Right. So rDNA technology, or genetic engineering, in other words, they have various applications. So it can be used to produce vaccines. It can be used to produce growth hormones, antibodies, and vectors. So, for example, in case of vaccines, we have the mRNA vaccine. So, or if you are having the spike protein, or the DNA vaccine. These all are the rDNA applications. Even in case of SARS-CoV-2, we had that kind of thing. Growth hormones are given, yes. Even in case of plants, it's mostly given. Antibodies to, let's say, snake bites, or a lot of toxins, venoms, these kind of things, they are produced using rDNA technology. So usually, then the vectors, for example, the plasmids, like I talked about, PGL3, PBR322, these are some very well-known plasmids which are used. These are also produced using rDNA. Then recombinant proteins like human insulin. There is a case study in the lecture 3 which talks about insulin. So recombinant proteins are produced using rDNA technology. Then anti-cancer drugs. Definitely for diagnosis as well, rDNA tech can be used, like gene therapy, which is present in this particular course. CRISPR also will be talking about it. Then monitoring devices and therapeutic strategies. Genetically modified food products like fruits, vegetables, crops, microbes, animals. So these are all genetically modified organisms called GMOs. There are various ethical concerns with this, and there are some energy applications also, like biohydrogen, bioethanol, biomethanols, which are produced using fungus, algae, stuff like that. So ideally, a normal fungus or algae will not produce bioethanol, but using recombinant DNA technology, we insert the gene and then we produce it. So this is some of the applications. Then we have the discovery. Okay, sorry. The discovery of insulin, and insulin was discovered as earlier when people had type 2 diabetes. It was not, people were not able to cure it. So, but later, from pigs, the pancreas of the pigs which were slaughtered, they were able to isolate insulin, and this insulin molecule was able to control the blood glucose levels, right, in case of humans. But since this insulin was coming from a different host, a different species of animal, there were several complications related to it. But now, scientists have developed methods to produce insulin in bacteria as well, so, or in eukaryotic cells as well. So they can do it using recombinant DNA technology approaches. You can read about it. So then there are BT plants, like BT stands for Bacillus thuringiensis. BT plants which have crytoxins. These crytoxins are important as they prevent the life cycle of pests, basically insects. So that's the idea. So we'll talk about the questions very briefly, and if you want to have interactive session, just before just for question number 1. So the question number 1 is, who coined the term gene? So you can answer this in the chat or Q&A, like you try answering it. If you know the answer, we'll discuss the answer for this. Yeah, the question is visible, Rakshit. So you can write the answer, whatever option you think is the right answer. You can write the answers, and we'll be discussing the answer very soon. Who coined the term gene?
So my chat option is not working. Can I speak like that?
You can speak. It would be better if you write it at Q&A.
Sir, my chat option is not working.
So like Shreya has already written, it's Q&A section beside the chat. So people are saying option B.
Sir, how to access that Q&A section?
Well, what is the device that you're using?
Sir, I'm on my laptop.
So in my laptop, I can see the Q&A section. I don't know if it is visible to all of you. It's just beside the chat. There is Q&A, a question mark. Okay, option B. So we all know that father of genetics is Gregor Johann Mendel. He had an extensive pea plant experiment, which was used to find out what were the laws of inheritance. Now, the term gene was, as you rightly pointed out, a lot of you, was given by Wilhelm Ludwig Johansen. Before that, Mendel used the term factor for describing this, gene. Now, question number 2. Histones are highly basic proteins which are found in A, eukaryotes, B, prokaryotes, both eukaryotes and prokaryotes, or D, plant. So you can give your answers. Okay. Right. So the answer is eukaryotes. We already talked about it, about the genome organization of eukaryotes and prokaryotes. So in case of prokaryotes, there are some protein molecules, RNAs, which are also present, but there is not histone in particular, which is present in prokaryotes. In eukaryotes, the genome organization of DNA essentially involves histone proteins. So eukaryotes would be the answer to this. Okay, this is the material behind it. You can study about it. So as I told you, there are like 4 histone proteins. Sorry, 5: H2A, H2B, H3, H4, and H1, and they form each individual nucleosome, and in each nucleosome, a DNA is bound around 1.65 times, right? Most prokaryote and eukaryotic chromosomes are both linear, both circular. Prokaryotics are linear and eukaryotic circular, or prokaryotic circular and eukaryotic linear. So what would be the answer? Okay, Gopinath said B. Jay Karne said D. Rutuja said A. I'm not sure if this A is for the previous one or this one. The answer would be prokaryotic chromosomes are usually circular and eukaryotics are linear. So that's the idea. That's usually how it is. There could be exceptions. Yeah, D is the option. So there is a difference table which is already present in the lecture slides. So prokaryotic chromosomes, they usually contain a single circular chromosome, whereas eukaryote, they contain multiple linear chromosomes. You know about it. So even in humans, we have linear DNA. So talking about question 4, first recombinant DNA molecule was created by A. Herbert Boyer, B. Stanley Cohen, C. Paul Berg, and D. Warner Arbor. So this is very important about the course. There are a lot of scientists and the years which you have to remember in this course. Okay, what are the answers? So people are having mixed opinions. C. Paul Berg would be the answer. So he created the first recombinant DNA molecule by combining the recombinant DNA from monkey virus SP40 with that of a lambda virus. So this is the cloning, step how he did it. So Herbert Boyer and Stanley Cohen, as we already discussed, they created the first transgenic organism by introducing antibiotic resistance genes into the plasmid of E. coli. And Werner, Arber, and Matthew Messelson in 1960s, they study about the type 1 restriction enzyme. Question number 5, type 2 restriction enzyme HIN2, was first identified and described from A. E. coli, B. Haemophilus influenzae, C. Salmonella typhi, or D. Bacillus subtilis. What would be the answer? I'll wait a little bit for others to answer as well. I think not everybody could access the Q&A. Okay, so the answer is Haemophilus influenzae. I'm sorry. Answer is Haemophilus influenzae, and this is how it is usually the nomenclature of enzymes are kept. So the first one is for the genus H, Haemophilus influenzae. The strains, we get the first two letters, and then the species, and then we use the strain, and then the order of discovery, which could be 2, 1, 3, whatever. So that's how enzymes are, named. So this is an example. For example, BAMH1 was isolated from Bacillus amyloliquefaciens, and EcoR1 from E. coli, PST1 from Providentia stuarti. These are some examples you can study about it. Type 2 restriction enzyme has 2 identical subunits, 2 different subunits, 3 different subunits, or only 1 subunit. What would be the answer? Okay. So type 2 restriction enzymes, they have 2 identical subunits. We already had read about the table. Type 2 restriction enzyme, they are unifunctional and they have 2 identical subunits. So that's for question number 6. Question number 7, M3 bacteriophage vector contains double-stranded filamentous DNA, double-stranded circular DNA, single-stranded filamentous DNA, or single-stranded filamentous RNA. Bacteriophage it has single-stranded filamentous DNA. So we know that there are 2 main important bacteriophage, the lambda phage and the M13 phage. So these they infect bacteria, right? And M13, they have single-stranded filamentous DNA. This is the structure of M13 phage. So crytoxin is produced by A. Bacillus thuringiensis, B. Bacillus subtilis, C. Pseudomonas aeruginosa, or D. Escherichia coli. What would be the answer? Would you like to guess? I am not receiving answers yet for this question. Okay, so BBA. So the answer would be Bacillus thuringiensis. Cry toxin is also called the BT toxin because it is produced by Bacillus thuringiensis. So we have already discussed about it. So they have these this toxin crystals, which are essentially toxic to insect species in the orders of Lepidopterans, Coleopterans, Hymenopterans, Dipterans, and nematodes. These are all pests which infect the crops usually. That's why BT cotton, BT brinjals, they have been produced to prevent these plants from these pest infestations. BT plants are designed to control A, lepidopteran pests, B, weeds, C, bacterial infection, D, fungal infection. We already discussed about it. So the answer is A, lepidopterans. We already discussed it in the previous slide. Right. Yeah. Question number 10. OREC is replication origin, transcription origin, translation origin, or cell division origin. Yeah. Anybody would like to speak something? I heard somebody switching on their mic. Okay, I'm getting answers. A, replication origin. Yes, you're right. Origin of replication is ORIC. So in case of plasmids, there is this origin of replication, which is very important for this plasmid to maintain itself in a cell. It's to replicate its own DNA, essentially. Complete set of genetic information in an organism is called a DNA, B, nucleus, C, gene, or D, genome. What would be the answer?
Genome.
Yes, genome, right. So we already discussed about it in the first slide itself. DNA is essentially is the material which we are talking about. It's present in the nucleus. Gene is the set of, it's the part of the DNA or the chromosome which has some function. So that's gene. And then there is genome, which is the entire set of genetic information in an organism. Question 12, who introduced the concepts of phenotype and genotype?
Ludwig.
Sorry, I didn't hear you.
Sir, is it Johnson?
Yeah, it is Johansson. Yes, right.
Right. Okay.
So for this, you need to study about the slides, about the lecture slides. I'm not going for it. What is the term for a single change in DNA nucleotide? Is it replication, transcription, translation, or mutation?
Mutation, I guess.
Yeah, mutation. Right, correct. So you guys have been doing pretty well. I'm getting answers from Q&A as well. Right. Question number 14. Which of the following locations do gene occupy as sequences of DNA arranged in one after another? Is it the cell wall, cytoplasm, endoplasmic reticulum, or the chromosome?
Chromosomes.
Right, you're right. It's the chromosome. Question number 15. The first GMP, GMP stands for genetically modified plant, was produced in 1982, which was, what was it? Was it BT cotton, BT brinjal, tobacco? Okay, yes, tobacco is the answer, right? It was first, you know, produced in 1982 and was used in 1992 by China. It was commercialized. By using DASH technology, insulin can be produced. Is it recombinant RNA or recombinant DNA, nanotech, or bioremediation?
Recombinant DNA.
Right, you are correct. The rest of the options, there's nothing as recombinant RNA technology, but there is RNA, a lot of RNA work which involves, you know, silencing, gene silencing, regulation, and all those kind of things. Nanotechnology is usually it's about nanoparticles, which could be metal nanoparticles, it could be liposomes, or other things which are used for usually drug delivery. And bioremediation means, you know, that is the concept of uh, in case of industry or in wastewater, you know, treatment plants, sometimes microorganisms are used to lower the concentration of toxins like cyanide and other pollutants are there. There are various petrochemicals, hydrocarbons. They are usually, you know, treated using microorganisms. That is called bioremediation. So these are the other technologies, but recombinant DNA technology, also known as genetic engineering, is used for producing insulin. Okay, then we have, this is probably the last question. Restriction exonucleases catalyze hydrolysis of terminal nucleotides from the end of DNA or RNA molecule, either in dash direction, 5' to 3', or 3' to 5', or is it 3' to 6', or 6' to 3' direction, 5' to 3' direction only, or 3' to 5' direction only?
Option A.
Right, option A. So as I already said, DNA, sorry, I'm not sure how I'm drawing it. Yeah, so DNA has two strands in anti-parallel orientation, 5' to 3', and then 3' to 5'. So in either of these places, exonucleases work and it cleaves this DNA. So that is all the questions that we have for today. Do you have any questions, any doubts? Now is the time you can ask. I'll just have the attendance sheet. Yeah, it's there. Any questions from your end regarding this lecture or for the course? Anyone would like to introduce themselves? I talked about myself. If you would like to talk about yourself, I'll get to know my audience better. So let's do it. Today is the first class. Let's ask each of you, where are you from and what are you pursuing currently? So we'll start alphabetically. I think Arya is the first one. You could introduce yourself.
Good evening, sir. I'm
Yes.
Arya Miss from Trivandrum, Kerala. I'm
Yes.
currently pursuing BSc Biotechnology, first year student.
Okay. From where?
from National College.
Okay. Okay. Fine. Thank you. Brijesh. Yeah, probably he's not available. Diksha.
Good evening, sir.
Yes.
I'm from JSS College, Dharwad, Karnataka,
Okay. Okay. And what is
and
your
I'm
degree?
pursuing BSc final year. BSc final
PhD.
year in
Okay.
biotechnology.
PhD
BSc.
final year. BSc.
Ah,
Okay. Okay. Fine. Uh, then the next name, Divyani.
Good evening, sir. So I am from Jammu, and
Right.
I am currently pursuing MSc in Human Genetics from Jammu University.
Okay. Great. Great. What was your BSc subject? What was your BSc
So my
on?
BSc subjects were biotechnology, zoology, chemistry, and English.
Right. Right. Okay. Dhanraj.
Good evening, Anna. This is Dhanraj from Tamil Nadu. I did my UG in BSc Honors Agriculture from Annamalai University, Chidambaram, and I've appeared myself for the PG entrance exam ICAR, and I'm awaiting for the results to pursue PG in agriculture.
Okay. Great. Great. Interesting. Gopinath.
Hi, good evening, sir.
Yes. Good evening.
Yeah, my name is Gobinath, and
Yes.
we completed a MSc Medical Biochemistry and MSc Computer Science. And I'm working in UAE as a clinical scientist for last 10 years.
Okay. Okay. Interesting. Interesting.
Thank you. Thank you.
Okay. Hajira.
Good evening, sir.
Yeah.
Myself Hajra Shireen from
Okay.
Tamil Nadu. I'm
Okay.
currently pursuing BSc Microbiology second year in
Okay.
Gandhigram Rural Institute, Dindigul.
Okay. Interesting. Jai.
Good evening, sir. It's actually Zai Karni.
Okay. I'm sorry.
I'm from Maharashtra and I'm currently pursuing my final year of BTech Biotechnology in Siagat College, Pune.
Okay. Interesting. Fine. Then we have Jakint J.
Hello, sir.
Yes.
Good evening, sir.
Yes.
My name is Jakint, and I am from Chandigarh. I am currently pursuing MSc first year bioinformatics.
Okay. From where?
From PU, sir. It's ST College under Punjab University, Chandigarh.
Interesting. Fine. Khushi.
Good evening, sir. I am pursuing in MSc Biotechnology and I am from UP Hathras.
Okay. Piyush.
Good evening, sir. So myself Piyush Rajput. I'm pursuing BTech Biotechnology second year from Pune Siagat Institute.
Okay. Pooja. Pooja, I think, is not available. Rakshit.
Good evening, sir. I'm Rakshit Shroff. I'm pursuing my final year in BTech Biotechnology from JSS STU Mysore.
Okay. Okay. And Rutuja.
Good evening, sir. I am Rituja Bhaviskar and I am pursuing BTech Bioengineering. I'm a first year student from VIT Bhopal.
Okay. Shreya.
Good evening, sir. I'm Shreya, and I'm pursuing BSc in Biotechnology from Techno India University in Kolkata.
PhD.
BSc.
Okay. BSc. Okay. They have a BTech and an MTech, I think, as far as I remember.
Yes, they have BTech as well.
I did my BTech from Heritage Institute of Technology, Kolkata. So anyway, T Dhar.
Good evening, sir. Can you hear me?
Yes, you're audible.
Yes, sir. I am Shrota Dhar, and I'm currently pursuing BTech in Biotechnology from Holdia Institute of Technology, third year.
Right. Okay. Yajurvi. Okay, probably she's also not here. So yeah. Okay, you're here.
Hello, all my
Yes.
fellow men. So I'm Yajurvi Mistry from Gujarat. Currently, I'm pursuing my bachelor's in botany, and I'm in third year. And I'm pursuing it from BKM Science College, Valsad.
Fine, interesting. So most of you are having some knowledge, as far as I can gather, about this subject, at least about molecular biology, about what DNA is, RNA is, what proteins are, and we'll be discussing in depth later on in the next weeks. And I hope it was an interesting session. If you have any feedback for me, please, now is the time you can tell me. If you want to change something, if you found something difficult, you can ask me right now. If not, you can leave and we can call it a day. I'll upload the recording as well as the slides.
Thank you, sir.
Thank you.
Sir, could you like reintroduce yourself? I missed that part.
Okay,
Actually.
so yeah, I'm Sanket Mukherjee. I'm a PMRF scholar from IIT Delhi, from the School of Biological Sciences. So my work, I'm in a molecular virology lab under Professor Vivekanandan Perumal and Professor Manoj Menon here in IIT Delhi. So I did my master's in biotechnology, MTech from IIT Guwahati, and in 2022. And I then started my PhD here in 2022 itself. And I have my BTech in biotechnology from Kolkata, from Heritage. So that's all about myself. So I had a gate rank of 179, gate BT rank in 2020.
Awesome. That's great, sir. Thank you.
I hope I'll see you all again next week. Thank you all for joining.
Thank you, sir.
Thank you.